So now that we're proceeding into chapter 15, we're going to be talking about the second half of protein expression, which is translation, and the mechanism behind that entire process. So keep in mind that because we're talking about translation, we're actually starting with the messenger RNA that was created in the transcription process, or that we went over in chapter 13. And this mRNA is going to go through a series of translation processes, and we're going to end up making a protein or a polymer. So there's quite a bit of information associated with chapter 15. I've got it broken down in about 5 main sections. So first of all, we're going to just do a brief overview of why translation is so important. Then we'll get into the structure and function of amino acids, how amino acids are created based on the nucleotide sequence. Then we'll get into the mechanism of transcription, followed by some of the modifications that can actually affect the protein expression. So, an interesting story that was actually in the first part of your chapter was about a condition that is a ribosomalopathy, where individuals that are or infants are actually born without a spleen, and a lot of a lot of times, folks that are older that lose a spleen because of some sort of accident, trauma, or whatever, they can actually survive without a spleen as long as they get through the infancy stage. But if they don't, and these infants are born without a spleen, they could really have their lifespan shortened, because the spleen is a key component in storing blood and filtering out bacteria in old blood cells. So just think about little babies and how often they get sick. And my niece and my nephew get sick all the time because they're in daycare. So if they didn't have a functional spleen, their lifespan might be at risk. So this condition was studied in 2013 using a combination of quantitative and molecular genetics. And what they ended up finding was that 23 individuals that had this condition, they compared their DNA genomes to about 500 individuals with normal phenotypes for this condition, or that had spleens intact. And what they ended up finding was that the RPSA protein was actually not being synthesized. And so, without that synthesis occurring, then that is one out of 33 proteins that actually come together and make up the small subunit of the ribosome. And that's going to become more apparent as we work through chapter 15. So this protein just didn't exist. Therefore, the ribosome wasn't complete and couldn't function at full capacity. So what they ended up finding was that a lot of these individuals with ICA, or isolated congenital asplenia, they had mutations in that RPSA gene. But they were still able to function. So that really told the scientists that there's many other genes involved in that disorder as well. So researchers ended up finding several types of mutations within that RPSA gene that were associated with the phenotype ICA. And one of them actually caused a premature stop codon to be synthesized. So that halted translation before the functional protein could actually be made. And it was interestingly enough a one frame shift mutation, and that'll become important as we work through chapter 15 as well. So this is a really good example of a phenotype that we knew existed out in society, but we were able to actually track down the cause of this genetic abnormality by using a combination of quantitative genetics to identify what gene was being affected, and then molecular genetics to actually determine that it was a premature stop codon. So they now have the ability, now that they know what it is, to try to work on some gene editing technology to try to prolong the life and the function of those individuals. All right, so chapter 15 is all about making proteins. And proteins are really important for a lot of different reasons. Proteins can be in the form of enzymes, they can be in the form of structural components like collagen or bone or hair. They are also going to be really important for transporting substances in our bodies. So, a lot of the hormones that are actually floating around in our bodies, whether it's thyroid or reproductive hormones, they actually are hitching a ride and are protected by proteins that serve as carriers within the blood. And then there's going to be a lot of proteins that are going to be used for regulation, communication, or some sort of defense mechanism like keeping us from getting sick. So proteins are going to be made up of amino acids. So an amino acid is a single building block of protein. So there are 20 different amino acids that make up all protein, and they work in a coding system. So you may have seen one of these figures before. I hope so, actually. So the way that these charts work, and you're going to see another one a little bit later on, is that amino acids are coded for in a codon, or a 3 nucleotide sequence. So the way that we actually identify the different amino acids is by that codon sequence. So the first position can be either U, C, A, or G, as can the second position and the third. So if we get an amino acid that is A, U, and G, that is going to be methionine. So AUG lines up with met, and methionine just happens to be our start codon. So there's 20 different amino acids that come together and make up all of the protein that is needed in our lives. Amino acids are going to be joined together by peptide bonds, and they're going to form polypeptide chains. So these individual amino acids have 4 major components to them, actually 5 major components. They have a central alpha carbon in the center. They've got an amine group, a hydroxyl group, a carboxyl group, and then they have a radical group, always designated as R. So the amine, hydrogen, and carboxyl group, in addition to the central alpha carbon, will always be the same for every single amino acid. The thing that changes is the side chain or the radical group. So amino acids are going to be joined together by peptide bonds, as we previously stated. So up here we've got one amino acid, and up here we've got another amino acid, and as you can see, our carboxyl group along with our central alpha carbon and our amine group are all present, and our radical group is also present as designated by R1 or R2, indicating 2 different amino acids. So this could be like methionine and arginine. They're going to be joined together in the polypeptide chain by this peptide bond here. So the carboxyl group specifically is going to be of one amino acid is going to be covalently linked to the amine group of another amino acid. So carboxyl group on one amino acid is going to be linked to the amine group of the other amino acid. Just like our DNA structure, amino acid structure can take on 4 different bigger structures. First of all is the primary structure, which really refers to the components that come together and make up the amino acid and how the amino acids are attached together in the polypeptide chain. Secondary structure really focuses on the interaction between amino acids and how they actually come together, conform, and twist into a helix to stabilize themselves. So that has to do with charges. Tertiary structure is how we see the primary folding of the secondary structure taking place. And quaternary structure is where we have subunits or different strands of protein actually coming together and fitting very uniquely so that that protein can be turned on or turned off. The amino acids, or the 20 amino acids, can be broke into 5 different groups, and I'm not going to spend too much time on these. I don't expect you to memorize them, but I do expect you to know what the 5 groups are. The first group is a non-polar aliphatic R group. Okay, so we're not going to have any charges associated with the R group. It's non-polar. The second group is a polar uncharged R group. The third group is where we start to see ring structures or aromatic R groups, indicating that the radical group is going to have at least one aromatic group. So these are going to be phenylalanine, tyrosine, and tryptophan, and these become very important in the function of some of our livestock and plant species because they tend to be so volatile as well. The fourth major group is a positively charged R group, so we can see that we're starting to get charges down here on our amines. And the fifth and final category is negatively charged R groups. So these negatively charged R groups are really important because they're going to have the negative charge off of the carboxyl at the very end of the R group. And these are only going to be aspartate and glutamine. And these become very important because they actually signal a functional protein. So a lot of times you will see aspartate or a glutamate, and sometimes even arginine, even though it's not negatively charged, actually come together and are going to be the start of a protein or a functional protein. All right, so we will pick up in the next section with how these amino acids actually work.